There is a fact about Native American blood that almost nobody outside of population genetics knows, and once you hear it, you cannot unhear it. Among the indigenous peoples of North and South America, the Navajo, the Cherokee, the Lakota, the Inca, the Maya, the hundreds of other nations and peoples who inhabited this continent before European contact, blood type O is almost universal, not common, almost universal. In many indigenous communities of South America, 100% of the population has blood type O. In most others, the figure is above 90%. Blood type A exists in small amounts in some North American groups. Blood type B is essentially absent across the entire continent. This is one of the most extreme genetic patterns in the entire human species. No other major population on Earth shows anything like this level of blood type uniformity. In Europe, 4 blood types are all present at significant frequencies. In Africa, 3 are common. In Asia, all 4 are found. But in the Americas, before European contact, there was essentially 1. Why? The answer takes us to a time when the world looked completely different. When a landmass that no longer exists connected 2 continents. When a small group of people survived conditions that should have killed them. And when, through a combination of chance and biology, they carried a genetic signature into a new world that would define the blood of their descendants for 15,000 years. This is not just a story about blood types. It is the story of the last great human migration, the final chapter in the peopling of the entire planet. Let us start with the data, because the data is extraordinary. A comprehensive 2026 study published in Nature confirmed what smaller studies had been showing for decades. The genetic diversity of indigenous American populations is significantly lower than that of populations on any other continent. Blood type O frequencies in indigenous South American populations range from 70% among Andean groups to 100% in Amazonian populations. The Yanomami of Venezuela and Brazil, one of the most studied indigenous groups, show blood type O frequencies approaching 100%. The Zavante, the Kaingang, the Ticuna, group after group shows the same pattern. O dominates, A is rare, B is absent. Among North American groups, the pattern is similar but slightly less extreme. The Blackfoot of Montana show high O with some A. The only significant exception is found among Arctic populations, the Inuit, Yupik, and related groups of Alaska and Greenland. These populations show more genetic diversity, including the presence of blood type B. This exception is a clue to how the Americas were peopled. To understand why, we go back 20,000 years to Beringia, a landmass larger than Texas that connected Siberia and Alaska. Ancestors of indigenous Americans spent thousands of years in isolation here. This period is called the Beringian standstill. This prolonged isolation had genetic consequences visible today, 15,000 years later, in the blood of their descendants. The key to understanding the blood type pattern of indigenous Americans is the concept of a genetic bottleneck. A bottleneck occurs when a population is dramatically reduced in size, and the reduced population then expands again. The genetic consequences are predictable and lasting. The expanded population carries only the genetic variation that happened to be present in the small surviving group. The ancestors of Native Americans went through one of the most severe genetic bottlenecks in human history. The population that eventually entered the Americas and spread across 2 continents descended from a founding group that was likely very small. Genetic analysis suggests this group may have been as small as a few hundred to a few thousand individuals. Every indigenous American alive today traces their ancestry back to this tiny founding population. The blood type consequences of this bottleneck are now well understood. The founding Beringian population, by chance or by selection, had very high frequencies of blood type O and very low frequencies of types A and B. When this population expanded across the Americas, it carried those frequencies with it. The expanding population remained reproductively isolated from other human groups for thousands of years. Oceans on three sides and ice to the north kept other humans out. So blood type frequencies were maintained and intensified over time. Scientists have identified a specific blood type O variant called O1V542, or the Native American O allele. It is separated from the common Asian O allele by a single mutation at a specific position in the ABO gene. This variant appears to have arisen during the Beringian standstill period, serving as a molecular signature of the founding population. Finding this specific variant in a blood sample is like finding a genetic fingerprint of the founding migration. The founding bottleneck explains why blood type B is essentially absent in Indigenous Americans. It was underrepresented from the start. But the near-complete dominance of type O in South America requires an additional explanation beyond just chance. Two mechanisms have been proposed: disease selection and competitive advantage. Before European contact, the Americas were free of several Old World diseases like malaria and plague. The disease landscape was different, so selection pressures on blood types were different. After European contact, the landscape changed catastrophically as smallpox arrived. Estimates suggest 50 to 90% of the indigenous population died in the following century. Blood type A is associated with increased susceptibility to smallpox, while type O individuals survived at higher rates. This means the already high O frequencies were pushed even higher by the epidemics. Modern blood type patterns are not just a product of the bottleneck, they are also a genetic scar of the colonial catastrophe. A 2023 study proposed that blood type O may have conferred competitive advantages in specific environmental contexts. Type O individuals may have had different immune responses to parasitic infections in the Americas. The evidence suggests type O was not just preserved by chance, it was actively maintained by natural selection. For most of the 20th century, the story of how the Americas were peopled was told as a single event. One group crossed Beringia, entered the Americas approximately 15,000 years ago, and spread rapidly across both continents. The near-complete genetic uniformity of Indigenous Americans was taken as evidence of this single founding event. The picture is more complex than that, and a landmark study published in Nature in April 2026 has added a new chapter. This was the largest genomic dataset of Indigenous Americans ever assembled, revealing a story that nobody anticipated. The 2026 study confirmed that the vast majority of Indigenous American ancestry does trace back to a single migration 15,000 years ago. After entering North America, these early populations expanded rapidly, reaching South America within a few thousand years. Around 9,000 years ago, a second wave of migration partially replaced earlier groups in some regions. This second wave is visible in the genetics of some North American populations and explains some of the variation in genetic markers. But the truly new finding is the identification of a third migration wave, approximately 1,300 years ago. This occurred when populations moved from Mesoamerica into South America and the Caribbean. This migration is visible as a genetic signature in present-day South American populations and in ancient Caribbean remains. The Americas were not a static landscape. Indigenous peoples continued to move, trade, and mix for thousands of years before European contact. Crucially, none of these subsequent waves brought significant amounts of blood type B into the populations they joined. The B allele was simply not present in the founding Beringian population in any significant frequency. The blood type O dominance is therefore a genetic legacy of the original founding event, rooted in that small group who waited in Beringia. The exception to the blood type O dominance is found in the Arctic populations, the Inuit, Yupik, Aleut, and related groups. These populations show more blood type diversity, including the presence of blood type B in some communities. Understanding why requires understanding that the Arctic was peopled differently from the rest of the Americas. The ancestors of the Arctic peoples did not come through the original Beringian founding population in the same way. They represent subsequent migrations from northeastern Asia that occurred after the initial peopling of the Americas. They crossed the Bering Strait by boat rather than by land, bringing with them different genetic profiles and blood type frequencies. A major study found that ancient Paleo-Eskimo populations made significant contributions to modern Arctic populations. These ancient populations had genetic profiles distinct from the founding Native American population. The Inuit and Yupik are not simply northern branches of the same founding tree as the Navajo or the Maya. They are descended from later waves of Asian migration that arrived by sea, with different genetic starting points. This distinction between land-based and sea-based migrations is one of the most important findings of recent research. It turns the story into a layered narrative of multiple migrations, multiple peoples, and multiple genetic legacies. The blood type pattern of indigenous Americans today is not identical to what it was before European contact in 1492. The centuries of disease, conquest, and population collapse that followed have left their mark on the genetics of surviving indigenous populations. The population collapse following European contact is one of the largest demographic catastrophes in recorded human history. Estimates of the pre-contact population range from 40 to 100 million people. Within a century, it was reduced to 5 to 10 million. The overwhelming cause was epidemic disease, smallpox, measles, and typhus, to which indigenous Americans had no prior immunity. This catastrophic reduction was a genetic bottleneck superimposed on the original Beringian bottleneck. The survivors had better resistance to the introduced diseases. Blood type O appears to have conferred some degree of relative protection against smallpox compared to blood type A. The survivors were therefore, on average, more heavily weighted toward blood type O than the pre-contact population had been. A 2008 study in PubMed examining ancient Native American DNA specifically addressed this question by looking at ancient remains. Ancient DNA before contact shows more blood type diversity than modern populations, suggesting the epidemics pushed O frequencies even higher. The blood we measure today is the blood of survivors, people whose ancestors made it through one of the most catastrophic disease events. Their genetics bears the mark of that survival as surely as it bears the mark of the Beringian founding thousands of years ago. The story of Native American blood types is a story about what it means for a small group to survive extreme circumstances. Those who lived in Beringia during the last glacial maximum had no way of knowing they were following game and moving with seasons. They carried particular blood type frequencies. When the ice retreated, their descendants spread across 2 continents, carrying those frequencies with them. 15,000 years later, a geneticist can look at the blood of a Yanomami person in the Amazon or a Maya in the Yucatan. The signature of that ancient founding event is written in the molecular structure of the ABO gene. The absence of type B is a record. It is a record of a journey across a vanished land, of survival through unimaginable hardship, shaping the biology of an entire continent. The blood of modern indigenous Americans carries the memory of both the founding and the catastrophe of colonization. The blood type pattern is a document written in molecular biology, recording events that happened before anyone was writing history. Subscribe to Hidden Origins, because the real story of who people are is always written deeper than history can reach. Drop a comment. Did you know about the Beringian standstill? And did it change how you think about the peopling of the Americas?